You said. This is Dr. Richard Land joining us from down the street, Cincinnati Children's Hospital.
Well, first of all, let me thank Elka and everybody else for pulling me into this fascinating discussion. There are a lot of themes that have been dealt with today that we're working with directly. So I'm very excited to be here and to be part of this. This is just a reminder about where we live. And as we've already heard, this daily pattern of EM radiation creates an amazing opportunity for the evolutionary process to generate adaptive pathways. So obviously this is the lighting in which we evolved over billions of years, but the lighting in which we choose to live is rather different. So we are, of course, cognitive. We think we know what we're doing. And so we have agency. And so we do things like this. Now, this is the total immersion room at a digital art installation at Artec House in Washington, D.C. This is an amazing creative event. And obviously, it's highly dependent on our ability to create and direct photons in particular ways. And I don't view this kind of event as any kind of a biological issue. This is entertainment, and this is fascinating, and it's just totally cool. But then we do things like this, where we create an entire city that has lighting at night. We do things like this. This is a close-up view of a cityscape. And then there is the ubiquitous electronic device that we stare at 24 hours a day. And so we've already heard from many of the speakers here that the lighting is coupled to physiology. And this is one of the big themes of our work right now. It's becoming absolutely clear that human physiology, mammalian physiology, is intimately coupled to the sunlight cycle. The way we live, we ignore that, and that's a problem. And there's this concept that we really choose to live in opposition to our evolved physiology. And so I'm going to tell you about some of the biology that we've been working on today, and also some of the biology that, of course, others have found before we got involved. This is obviously the Chris Palczewski crystal structure of rhodopsin. The rhodopsin class GPCRs are, of course, the major light sensors in animals. You can elicit a biochemical response in a cell when these molecules capture the energy of a photon. And we've already heard that different classes of opsins can absorb wavelengths within particular narrow ranges. The one that may be especially interesting for this discussion is opsin 5, that's on the far left of this. It's a short wavelength opsin. There are no photons in this room that would stimulate that opsin. We'll come back to that. And then, of course, excepting about the last 200 years, the primary source of photon ligands for these molecules was the sun. And so there are consequences for the way we choose to live now. So I'm going to tell you about the so-called atypical opsins, opsin 3, 4, and 5. Opsin 4, there's a whole field of inquiry grown up around this molecule, and it's absolutely fascinating. It's a blue sensitive opsin. It's expressed in retinal ganglion cells like those. You can see that image in the top right. We've already learned that it has a key role in circadian clock entrainment. And I want to point out also that our new faculty member at Children's Hospital, Diego Fernandez, who's sitting right over here, did some beautiful work a while back demonstrating that this opsin is involved in a pathway that regulates mood and learning. My group has been working on neuropsin opin 5 for a while. This, as I mentioned, is a near UV sensitive opsin. The peak absorbance is about 380 nanometers. We already know from the work of my collaborators Ethan Buer and Russ Van Gelder that it is involved in photoentrainment of circadian clocks, both within the retina and within the skin. And it's also involved in regulating seasonal breeding behavior in birds. That image you see there is showing a brain section through mouse where we've identified opin 5 expression. And that little patch of red in the bottom of the brain section is an area called the preoptic area. And we'll come back to what that does physiologically. Opsin 3, encephalopsin, is a little bit more of an enigma. It clearly falls into the opsin class of molecules. there's limited information about its light sensitivity because it's a difficult molecule to work with. But this movie you see down on the bottom is actually from my collaborator Leland and Hager, and it just shows that opsin 3 is expressed throughout the mouse brain. This is a day of birth mouse brain. And so this, then there is expression in white adipocytes. I'll mention what that does. And so it raises this very big question. Does this mean that all these tissues are light responsive? So my colleagues in the mammalian biology world have widely stated emphatically that mammals have lost their extraocular light sensing capabilities. So I was delighted to see that we had a lot of examples of extraocular light sensing in other species. That position is clearly wrong. There is extraocular light sensing in mammals, and it's going to be crucial for some fundamental physiology, as I'll show you. So circadian clock. So we know that there is a photoentrainable circadian clock that uses a light input pathway from the eyes that uses opium-4 or rod and cone photoreceptors. That is a pathway that photoentrains a circadian clock in the suprachiasmatic nucleus. There is another circadian clock in the retina that uses opium-5 as a light sensor. This is work from Tassini and Maneker and Ethan Buer and Russ van Gelder. This circadian clock functions entirely independently of the opium-4 SCN circadian clock. And then also from my collaborators, it's been shown that opium-4 in the skin, in melanocyte precursors in the mouse, runs a photoentrainable circadian clock that also functions entirely independently of the other two circadian clocks. So we now have documented at least 3 circadian clocks in a mouse, and it raises the question of how many there will end up being. So one of the developmental pathways we're very interested in the mouse eye is vascular development. And this schematic just summarizes how 2 light sensing pathways are involved in regulating that process. So this, the light sensors that are crucial here are melanopsin opium-4 on the left-hand side of the schematic and neuropsin opium-5 on the right-hand side. So in this instance, melanopsin functions locally within the retina to suppress vascularity. And this is light sensing that occurs prior to and just after birth in the mouse. On the other side of this schematic, the opium-5 response promotes vascularity, and this is light sensing that occurs after birth. So we look upon these pathways as developmental timing cues. All the evidence, the experimental evidence suggests that that is the case. And this is actually relevant to a rather obscure disease called retinopathy of prematurity. When a child is born premature, the change in oxygen tension that they experience puts them at risk of getting a dramatic vascular overgrowth in the eye. And if it occurs badly enough, it can drag the retina off the back of the eye, and they can be blind for life. So there's this very interesting possibility that we might use these light sensing pathways to treat that disease. So we already talked about myopia a little bit. This is a massive public health problem. Right now on the planet, there are about 2.5 billion people with myopia. And if we don't learn how to fix this problem by 2050, there's going to be 5 billion people. Minor myopia is not too much of an issue, but if you have a severe case, you're at much higher risk of a whole series of sight-threatening complications. And so we need to know how to deal with this. So a lot of work done by many groups over many years has shown that all the opsins are involved in regulating how the eye grows. If the eye grows too long, you have myopia. But most recently, what we found was that opsin 5 was involved in this process. So opsin 5 sensing of violet photons is crucial to actually prevent elongation of the eye. And so that establishes this really interesting causation argument that because we live inside and the windows prevent violet photons from coming in and the lighting systems don't make violet photons, we're living in an environment that is pro-myopic. And so there is this very interesting possibility that if we just change our lighting systems, we can prevent myopia. Or go outside for 2 hours a day, as Elkin mentioned. So, so this is a really interesting possibility. And the last physiological pathway I'll mention actually involves opium-5 and opium-3. And so work we did recently makes a case that that little patch of opsin 5 expression in the preoptic area of the brain is light sensing. And yes, you can show that photons can reach that part of the brain. And when that is stimulated by violet light, you actually get a sympathetic nervous system output to the brown adipose tissue that is specialized for the creation of heat. And this pathway actually suppresses activation of brown adipose tissue. And at the same time, we've shown that white adipocytes use opsin 3, or at least they have a light-dependent response. There's also dependent opsin 3 that actually enhances the production of free fatty acids through a pathway called the lipolysis pathway. And those two work together to give you the generation of heat within the body. And so the mutant mice, for each of these opsins, they have a body temperature phenotype, just to illustrate the point. So obviously what this is teaching us is that these light-sensing pathways are involved in regulating energy homeostasis. And obviously we argue it's going to be a circuit. There's going to be some kind of endocrine feedback that gives you homeostasis. But what these analyses do is identify a new mechanism for regulation of energy metabolism in which coupling to the light-dark cycle is a key feature. And so this slide just summarizes that mouse schematic there, just summarizes all the light-sensing components that we now know about. And I won't go through all of them, but you can see it looks like a mouse that is pretty well set up systemically to respond to light. And so I think as we learn more about all these light-sensing tissues, that's going to be the conclusion. And so when we had the opportunity to build a new hospital, Children's Hospital, it's called the Critical Care Building, we decided to take all this biology into account when we established the neonatal intensive care unit rooms. And working with this gentleman, Jim Greenberg, who is my clinical counterpart, when we put this hospital together, we decided to design a custom lighting system that had all the wavelengths to stimulate all these opsins, and was also a circadian lighting system. So this is the spectral power distribution of the lighting system. You can see that it's designed to stimulate opsin 5, opsin 3, and opsin 4, among the others. And so in essence, it's a circadian lighting system. It goes through a cycle every day, but it produces violet photons for the stimulation of opsin 5. And this is just a little illustration of how the user interface works. A user here is picking a particular power distribution from each LED channel. The way this works is that once you've chosen what you want, you can save that information as a static description of a spectral power distribution that we call just the keyframe. Then you can join those descriptions of spectral power distributions into a time course to give you a spectral day, and join days together to give you a full lighting protocol. And so obviously our goal here is to use this system on patients who might benefit from having a particular lighting protocol. And we're thinking about things like retinopathy of prematurity, that we already discussed, maybe stimulation of children who are susceptible, who are at risk for ROP will help. But also there are many patients in the NICU who have growth insufficiency for various reasons. And we are wondering whether using a particular lighting protocol, because of those metabolic pathways I've described to you, whether that might be beneficial for them as well. Some folks in the big wide world have started to take notice of this biology, and this is a paper that I wrote with these two individuals, Kevin Hauser, who's at Oregon State University, and Lisa Heschong, who is the most energetic retired architect I've ever come across, that really just asks the question of the building science community of whether the way we design buildings is causing a problem for myopia. And so obviously windows block violet photons. We don't have lighting systems that produce violet photons. It was just raising the question of whether that might be a problem. And so what will biologically aware buildings look like? Well, on the right-hand side, this is just a summary of all the light-sensing tissues I've mentioned. Buildings are on the left. So maybe what we can do here is generate glazing designs that actually allow more of the short wavelength photons in, and maybe we can have lighting systems that will create those photons in such a pattern that is beneficial rather than detrimental. And so, you know, there's this big question: can we persuade the glazing and lighting industries to come to the party? So this is my crew. They've done an amazing job with all of this biology. Jonathan Mercado-Reyes is here. Daniel Kang is here. And I think that might be it from my crew. We have a lot of fantastic collaborators. I mentioned Jim Greenberg, who's been instrumental in getting this work done. And I would just mention that, whoops, we have started a new program at Children's Hospital called the Science of Light Center. They're designed to get all of this new biological information out into the big wide world, as well as use it to advantage in the generation of healthy outcomes for our patients. So with that, I will stop. And if there are any questions, I'll be happy to answer them. Thank you. Okay.
Fascinating. So I'm particularly new for me the OPN3 and the whole metabolism of heat. That's fascinating because in invertebrates we have looked at opsin expression, and there's this opsin 7, which keeps being in this weird place. As my student Shubham Batoratek there found, it's in the port cells next to the photoreceptors instead of the photoreceptors there. And so I'm wondering, maybe, and so in order to see well, your photoreceptors also have to have a certain temperature, and invertebrates are in all sort of environments. So you're giving me this idea that maybe one of the functions to have the food production that would be, and also there's a very metabolically active cells. They're in charge of the energy that's being supplied to the neighboring photoreceptors. So
yeah, that's an interesting idea. I mean, the mechanism of action of OPN3 is really not understood yet. It's been very difficult to work with at the molecular level, and so we don't really exactly understand that it functions as a conventional opsin.
Yeah.
Nobody's done the sort of biophysics component of analysis yet. That will come.
But generally, there seems to be opsins popping up that are not immediately visual, or like they don't seem to respond to light, even though they still absorb light. The whole idea that could trigger something that has to do with metabolism, I think, is a fascinating.
Yeah, I would be very surprised if we don't see this kind of thing in many species.
Right.
Yeah.
In marine systems, lunar light is very important to viewing into lunar cycles, full moon, new moon. Do you know of any mouse research, even looking at that at all, or do you think it's possible that lunar light also matters?
Well, so not mouse research. So, the typical lab mouse is highly derived and not a good model for what happens in the wild. But there's some very recent evidence that came up at the Society for Research in Biological Rhythms that clearly showed in the human population there was a response to the lunar cycle.
On light, or not necessarily?
Well, I don't think we could say it's to light, but the pattern of activity in the human population was there.
But as a non-scientist, it seems like a number of people's research is around kind of like simulating the sun in some way. Is there any research into whether or not the scale of the sun, the distance of the sun, the way it kind of interacts with the Earth? Is it possible to simulate it in some regard? As I'm saying, would be this singular related to something? Is there something can you not possibly make a simulated sun in your space with scale?
I think it's a great idea. In fact, we at one point, in other words, is visual tracking of the sun across the sky an issue for physiology? I don't know the answer to that, but I think it's a fascinating idea. Actually, we went one step further a while back, and I persuaded the engineers we have access to at Children's Hospital to make me a lighting device that mimicked the sun in our mouse room. And it turned out to be fairly complicated to do that. And we didn't actually assess it, but I think it's a really interesting idea.
Sun simulators.
Say that
You
again.
can buy sun simulators.
Can you buy a sun simulator? You can.
Yes, they're on the market.
I'd love to take some measurements of those things.
Exactly, because sometimes you buy things and they say there's unless the spectrum actually checks out.
Yes, you can buy them. Specifically, they're supposed to simulate.
Did you hear that, Daniel? Buy one.
Can you speak to the genetic aspect between it, how much with these opsins are affected by the light versus how genetics are playing in, like, you know, at least it's general public been like an understanding that myopia has a genetic element. Like my dad is severely myopic, I am as well. We didn't grow up anywhere near each other, you know. And so it's just interesting to see that there could be this effect that you're having with the lighting, but how much the genetics contribute.
Yeah, so myopia is a great example to address this question because it's been so thoroughly studied. So there very clearly is a small genetic component to myopia. I mean, population-wide, the effect sizes are quite small. For any individual, of course, it's difficult to address that question. So, you know, your particular case of myopia could have a large genetic component. So, you know, the genome-wide association studies that have been done on large populations of people with myopia say that on average, the genetic contribution is small. And of course, the way myopia incidence has been accelerating, it's very difficult to argue that it could be a genetic disease. So you really have to argue that on average, it's an environmental disease. And that's why these light sensing pathways are so compelling, because it's an explanation for why this is happening.
Can the light also play a role in the genetic aspect of it? Like, is it a way of combating some of it? Like, I guess, can it affect that aspect of it?
Quite possibly, yeah. So, you know, this comes right down to the kind of experiments we do. So when you induce a myopia in an animal, you actually put an external lens that changes the way the light refracts into the eye, and you induce abnormal eye elongation. Now, it turns out that violet light can suppress that elongation even when you have the lens outside the eye still. Okay, so I think the answer to your question is probably yes. Thanks, everyone.